Hello and welcome to Science Research Weekly, Season 4, Episode 13, Genomes from Green Places. Get ready for Green Sahara genomes, super-duckweed, Alzheimer's blood tests, Volume 112 of the Journal of Statistical Software, a litmus test for R packages, and the top 40 new CRAN packages for February 2025. This episode covers material from March 20th to April 3rd. Let's get started with some research articles. First on the docket here is ancient DNA from the Green Sahara reveals ancestral North African lineage. The Sahara, the world's largest hot desert, was a green savanna during the African human period, ending around 5,000 years ago. Researchers found and extracted genomic data from two 7,000-year-old pastoral Neolithic females from the Karkori rock shelter in southwestern Libya. They found that their ancestry stemmed from an unknown North African genetic lineage that's no longer around. All right, shifting gears pretty dramatically, we have, though sticking with genomes, duckweed genomes and epigenomes underlie a triploid hybridization and clonal reproduction. Did you know that duckweeds are the world's smallest and fastest growing flowering plant? Well, like me, you do now. Looking at updated genomes from duckweed genera, so that's Lemna, researchers found answers on genes involved in these duckweed anatomical traits. Also, they found connections to events in evolutionary history and made the case for Lemna species as a powerful model organism for biotechnology applications. Like, for example, they were able to engineer them to produce like way more oil, per gram or per plant than conventional species. So, space agriculture with duckweed, I'm looking at you. Next article is Plasma MTBR tau 243 biomarker identifies tau tangle pathology in Alzheimer's disease. Researchers, they developed a blood biomarker, which is endogenously cleaved microtubule binding region containing residue 243, or EMTBR tau 243, from plasma, which accurately reflects tau protein aggregates within neurofibrillary tangles of the brain, which are a defining neuropathological feature of Alzheimer's disease. So hopefully with this blood test, can better detect it, especially early on, which is one of the problems with trying to do something like, say, treat Alzheimer's disease, if you aren't able to detect it early enough or accurately enough. So this is a promising blood biomarker. And that's sort of, for a lot of these diseases, having a blood biomarker or another easily detectable biomarker is sort of the holy grail, as it were. All right, let's turn to our next article here. Genome duplication in a long-term multicellularity evolution experiment. So unfortunately, this article is behind the paywall, but the abstract looks very intriguing. So researchers, they ran a long-term experiment. I think it's still running, actually, in a diploid snowflake yeast. So that's Saccharomyces cerevisiae. They found tetraploid strains that persisted for over almost 5,000 generations, despite the genomic instability that tetraploidy exhibits. The reason they persisted, there's a fitness benefit under a selection for larger multicellular size. So yeast mutants that got the tetraploidy kind of got an initial boost, and that was stable enough because the researchers were selected for a large size for that to stick around. And this research suggests that whole genome duplication can arise because of that short-term fitness benefits, but actually can convey long-term benefits, long-term innovations because of the added genetic variation. Because if you have two genomes, you can play around with it more, take more risks because you have a backup. And that's, I think, a big thing in the research of genome duplication is seems to be a common mechanism for new genes to come about when you have that backup of multiple DNA. All right, finishing out our time here with the articles, we have Cooperative hydrodynamics, accompany multicellular-like colonial organization in the unicellular ciliate Stentor. So researchers found that unicellular ciliates feeding in water together, and they have their cilia, they're beating it, they're sort of churning up the water for like a flow to bring small particles to feed on. They found that these ciliates feeding in water together reached a lot higher flow rates than individuals working alone. And this actually benefited all members of the aggregate. So suggesting that one of the possible driving forces of multicellular organization in the deep past may have been driven by this benefit of faster feeding currents when aggregated. So that was really interesting, sort of modern, sort of nature physics experiments, observations, sort of connecting to potential deep past events. All right, well, let's turn our time here to some research tools. First of all, the Journal of Statistical Software, volume 112, has started dropping with several issues already. These include GP tools, scalable Gaussian processes, interface with STAN, Resi, an R package for robust effect sizes, split, apply, combine with dynamic grouping, learning permutation symmetry of a Gaussian vector with GIPS in R, Pyriclet, a Python package for density estimation and clustering using Gaussian mixture models, and finally BaseMix, Bayesian mixture models in C++. Rick Wicklin from the Do Loop has two new posts, one historical, the other statistical. First, he answers the question, did George Box say all models are wrong but some are useful? Yes, he did, writing it as early as 1976. And he has, Rick Wicklin has excerpts to prove it. Second, he shows how to create, Rick Wicklin, that is, not George Box. Rick Wicklin shows how to create nested bar charts in SAS using the graph template language. He makes a note that about 95% of graphs in SAS can be created by 95% of the users using the simple SGS procedures in SAS. Those less common, the most complex graphs are gonna, that 5% remaining are gonna take a lot more effort just because that's what's needed, and that the graph template language is one way to do that. Alrighty, 3 new R bloggers post today. First, from Keith Goldfield, the finale post on Bayesian proportional hazards model for a stepped wedge design. Second, from the Jumping Rivers blog, should I use your R package? Which goes over the Litmus package validation framework to help people decide whether or not to use an R package from the standpoint of things like quality and risk. Like how secure is it? How documented is it? Things like that. And I think one of the specific examples is like for something like pharmacologicals, it really needs to be, packages really need to be well regulated, well secured, whereas something like a researcher maybe doesn't need that much sort of safety. And then third, from Joseph Rickert, the February 2025 top 40 new CRAN packages have dropped in a variety of different categories, from AI to weather. So a lot of interesting things to look at. There's like, for example, chores, providing a collection of ergonomic large language model assistants to help complete repetitive, hard to automate tasks quickly. I mean, there's things like Vijaya Kana implements a toolbox to perform analysis of the WHO pharmacovigilance database. there is discrete DLM, provide tools for fitting Bayesian distributed leg models to counter binary longitudinal response data. So a lot of interesting, possibly useful packages for you to explore more yourself. Speaking of new packages, new CRAN packages, I have, I believe, 5 for you today that I think you might check out that came out in the last week. First up is CastGen, estimate sample size for population genomic studies. There is classification ensembles, automatically build 20 classification models. The next two are in that line. There's forecast ensembles and numeric ensembles, both using a variety of models for either time series forecasting or just individual or ensemble models for numeric analysis. And then finally, there's PCPR, Principal Components Pursuit for Environmental Epidemiology. So with that, we get to research funding. There's no new funding of note. So I'm going to close off with something a little bit different, a dare. Check out the link in the description below to a working paper, 2017 by Bloom et al. The paper is called Are Ideas Getting Harder to Find? Figure 1 is a graph. And once I saw that graph, I could not unsee it. So if you're willing to risk the knowledge from that graph too, go on, take a look at it. The scales fall. Thanks for joining me for Science Research Weekly. Science on.